Hi Hires, so Metabolism and Survival, we are looking at recombinant technologies that can allow us to improve microbes. So one of the first things you have to be able to do if you're working with DNA is you need to be able to split it up. And by that I mean separate out your fragments. So what you often do is you smash up your DNA, or you've maybe done PCR on your DNA and now you've got a chunk that you want, but you've got all sorts of other bits mixed in with it. So what we can do is separate them using gel electrophoresis. And gel electrophoresis is a really nice technique to do this. So what we have is this basic setup. So in here you have a tank that's filled with a buffer. So this is a buffer in here. So that's a liquid that's going to allow everything to move. And then we've got it hooked up here to a power pack. And then inside this buffer, what you can't see particularly well in this one, but a little bit better in this picture, you have a gel. Now this gel here has basically been poured into a little frame and set. And then as you were setting it, you've also used something called a comb. So something that when you set it up, there was a little thing like this, slightly neater, that's been slotted in as it sets, and then you pull this out and you've got tiny little holes. And then each of these holes, now called a well, you can put in a little sample of your DNA. Okay, now DNA is not coloured, so this dark blue blobs that you can see here is because you put a dye in so that you can see what's going on. And then all you need to do is to hook up the charge. Now DNA is negatively charged, which you should know because you know that it's wrapped around positively charged histones when it's doing its packaging. So the negative charge means that when you push it, it gets pushed through the gel. So what's really important, essential, is that you hook it up the right way round, okay, because what you want is to have the negative charge at the top here with the wells, and that will push the DNA through. And actually it's the smallest bits that move fastest, and what you end up with is these bands. Now this is being shown up using a UV stain. This first one here will have been set up with something called a ladder, and so this will be particular batches of something that's already been set up. So it will be set up with something, say a 20 kilobase, a 10 kilobase, a 5, a 2, a 1, so that you've got all of the different sizes of DNA, so that if you know you want to pull one out that's say 11, and this is your 20 and this is your 10, you're like, in fact that won't be 11, this would be a 9, okay, smaller. So we can actually pull out that one, and you can look to see what bands we have. In this case, in this one here, they don't have the band there, so they don't have that particular bit of DNA. I like to think of it this way, okay, if this helps. So if you imagine a forest, very densely wooded, lots and lots of branches, thicket kind of level, okay, and then you imagine two different sizes of bird. We have a little bird and a big bird, okay. Now imagine that we put something which scares them. So imagine these are the little DNA and the big DNA strands, and we now bring in something negative, which they do not like to be near. So we bring in something scary. Okay, now think about little Tweety Pie. It will scoot through every single branch. It will just avoid them all, zip, 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 right the way through. Big bird, however, will get to here, trip over, fall, get back up again, smack into this bit, and basically won't get very far at all. So we end up with little band down here, big band up the top. Okay, so just to remember that we have it the opposite way around than you might think. Okay. Okay, another thing you need to be able to do, and another thing which, once we've learned how to do it, has revolutionized, and there's lots of times I might be saying that, biology and how it works. Okay, because what we have here are a group of enzymes, and there are literally thousands of them, that have been isolated from microbes, and they are enzymes, so that's an A's. They cut, so that's restrict, and it's inside a DNA strand, so it's endonucleic acid, hence restriction endonuclease. Okay, the name actually does make some sense. If you think about why that might be inside a particular bacteria, then if you think about this idea from the transduction, if you have a virus which has just injected you with some of its own nucleic acid that is going to take over your cell, having enzymes which will basically just chop it up are really useful. And we've basically co-opted that. And they're now named after the organism they originally came from. So restriction endonucleases are really useful because they find a particular sequence and then they cut it. Okay, now there are lots and lots actually of different endonucleases about how they do this. We're really only talking about one particular type, okay? And what they do is they cut in a particular sequence and they snip it across and down. So if this is the sequence for the enzyme BAMH1, so GGATCC, they cut it across and down. And what that means is you've got this end from the bit you cut, which is CTAG, and you've got this end, GATC. So these are sticky ends because they're ready for a complementary sequence to come in. So these could just join back up again, or if I got rid of this one and I just had this end open here, I could bring in something which had that GATC, so it'd been cut with the same enzyme, and it would stick on here. Okay, so you have a sticky end. Actually, really, you could have one that goes like that, might be a better way of looking at it. Okay, so we get the whole thing joined in. So when we've talked about genetic engineering and we've said cut open the plasmid and then insert, this is how the cutting opening is happening. Okay, and generally when we've drawn them, you might not have noticed it, but we were often drawing them like this. So when we cut open the plasmid, we were drawing it like you had an end and an end. So we had, there's a sticky end here, and here's a sticky end here. We're trying to show it, but only talking about it now properly at higher. So just to see that we have lots, these are all examples of restriction endonucleases, and these are the names. BAMH1 was the one that I mentioned on the last slide. So this is where we've got BAMH, and it's the first one we got, so BAMH1. Okay, PSD1, so P, ST, and it was the first one, 1, and you can see how they all kind of link up. And you can see here that we have different sequences, and you can see where they cut, and some of them have shorter sequences, some of them have longer, and there are even some that don't cut with a sticky end but create something called a blunt end. But you don't need to know huge amounts about them. So this is just a picture showing how BAMH1 might wrap itself around the DNA spiral. So here's your DNA spiral in blue in the center here, and these are the two units that make up BAMH1. And this is a particular way of modeling proteins. So what you have are these arrows showing something called beta pleated sheet, and then we've got things called alpha helixes going up the side here. And it's just a way to try and cut down how much space it's taking up. If you used a space filling model, so you showed every single atom that was involved here, the whole thing would be clamped round. But what's really impressive is that it is so precise. It's able to actually know the difference between a C, a T, an A, and a G when it's fitting round this DNA, and it will only clamp round perfectly with the right sequence. And if it isn't the right sequence, it just doesn't clip round. Okay. So far, you can now cut bits of DNA, you can sort them using gel electrophoresis, and now what's really important is you can now stick them together. But this is something you already know. You already know which enzyme we're going to use for this. We're going to use ligase. So once we've got our sticky ends, the right fragments, the things we need, mix them together, put in ligase, and we get our recombinant DNA. So assuming we get everything right, it's not the easiest thing to do, but the enzymes are all there. Our final DNA is, as I say, recombinant, taking all of these things that we're doing, restriction, separating, ligation, meaning sticking, you can get these modified plasmids, and you have now got DNA from different sources. Plasmids are great, but there is a limit to how much they can take in terms of the fragment size. So there are things that have been designed to take a bit more, and these are what's called artificial chromosomes. You can look up the ones for bacteria and yeast ones. Just thinking about it, okay, we're talking about microbes, but what would you put on a human chromosome 47? Okay, so looking at what you actually have inside a plasmid. So when we talk about a plasmid, you've just drawn a kind of grey circle or black circle or whatever colour you decided to draw it, and then you just cut it and you stick something in, and we've just said that's it. But there's more to a plasmid than that. Now, one of the most essential sequences you have to have is an origin of replication. So in this case we've got it down here. Okay, so the origin of replication means that once you get that modified plasmid into the cell will make extra copies of that plasmid. If you don't have this, then if you imagine a bacterial cell that has got one copy of the plasmid, when it divides, there's only a 50-50 chance of that plasmid going to one cell or the other, and that's not any help to you. So what you need to do is make lots of copies of the plasmid so that when the cell divides, you get all the cells that are coming out with this extra plasmid. Okay, so this is essential code. Okay, you also will have marker genes, which will help you select cells that have got the plasmid, and it will control what cells grow. Okay, we've got regulation sequences, and they're really important to control how much the gene that you've just put in is expressed, how often and how much. We have obviously need to have restriction sites, because otherwise you're not going to be able to cut and put the DNA in. And then we also have control genes, and these are genes that we've got inside the plasmid to control whether that cell will survive outside of the lab environment. So basically we want it not to. You want to control your modified organisms. Just to show you, this is a commercial plasmid, so this is one that you can buy to use in E. coli, and I just wanted to show you the kind of size that we have of this one. You'll notice that we've got our different genes that we would expect to have in here. So what we've got here are some resistance genes for different antibiotics, and every single one of these is a restriction site. These are all different restriction enzymes, and what you have is the code number here we have is just where it is, assuming they've decided on a start point, and then they just work their way around. Okay. So what can you actually do once you've got this system all set up? And frankly, it's a huge amount of different things. But we can produce transgenic organisms. They have proteins that we produced in other organisms and not originally in them. And that is pretty mind-blowing when you think about how that could actually be applied. You can control a particular product because you can influence the pathway that's being used, because you can get rid of inhibitors, or you can change metabolites, you can change what's going on inside the pathways. Now obviously you need to explain the pathways and describe them and know what's going on with them before you can do that. So assuming that you've managed to do the really hard stuff in terms of finding the gene you want, cutting it out, separating it, cutting it with restriction endonuclease so it's got the right kind of sticky ends, finding the right plasmid, cutting open the plasmid, getting it all stuck together in the right way around, and you've got it into a cell, you're not really finished there, because gene expression is really tricky to look after, and so you need to have too much or too little. That can be an issue even if you've got as far as having the gene in there. And then on top of that, you might have the right gene and it might be expressed, but it might not actually work as the final protein, because eukaryotic genes are treated a little bit differently inside eukaryotic cells. After they've been produced in translation, then proteins quite often go through another set of stages called post-translational modification, where you add things in, chop little bits off, finish off little folding. So prokaryotic cells struggle to do that with eukaryotic genes. How do we deal with those? Gene expression is all about those regulation sequences that you have, so trying to control the gene expression that way. Incorrect folding, basically we go for something that is eukaryotic already, so we go for a yeast cell rather than bacterial cells, and that means that we should get the right folding and modification afterwards. Okay, that's us.